Hi friends, in this video we have 15 questions on Operon concept, one of the most common topic in competitive exams in biological sciences. Let's begin with a very simple one. In prokaryotes like E. coli, what is the term for a cluster of structural genes with related functions grouped under the control of a single promoter called? Option A, enhancer. Option B, operon. Option C, intron. Option D, gene. You have 10 seconds to answer this question. The correct answer is operon. As you see, these are structural genes LAC-Z, LAC-Y and LAC-A. This is controlled under a single promoter that is called as operon. This is your scorecard. You can watch our video on LAC-operon before attempting this quiz, or let's move to the second question. Which of the following is not a component of an operon? Option A, promoter. Option B, operator. C, genes. D, terminator. The correct answer is terminator. Terminator is not a component of an operon. Question number 3. What is the function of the promoter in an operon? Option A, it binds repressors to inhibit transcription. Option B, it is a DNA sequence where RNA polymerase binds and initiates transcription. Option C, it encodes the structural proteins. Option D, it facilitates the entry of substrates into the cell. The correct answer is promoter is a DNA sequence where RNA polymerase binds and initiates transcription. So this is a promoter where RNA polymerase binds and initiates transcription. Question number 4. The operator in an operon is a DNA sequence that a repressor or activator can bind to. What is the consequence of this binding? Option A, it always enhances transcription. Option B, it always inhibits transcription. Option C, it can either allow or inhibit transcription. Option D, it has no effect on transcription. The correct answer is it can either allow or inhibit transcription. So this is the operator. If the repressor binds to this operator, it inhibits transcription. If an activator binds to this operator, it activates transcription. Question number 5. What is the key characteristic of the transcript produced from an operon? Option A, it is always monocystronic, encoding multiple proteins from a single mRNA molecule. Option B, it encodes only one protein. Option C, it's a polycystronic transcript encoding multiple proteins from a single mRNA molecule. Option D, it is immediately translated without further processing. The correct answer is it is a polycystronic transcript encoding multiple proteins from a single mRNA molecule. As you see here, this is the polycystronic mRNA transcript that is formed by the transcription of the structural genes encoding multiple proteins from a single mRNA molecule. Question number 6. Which gene in the LAC-operon encodes beta-galactosidase, the enzyme that breaks lactose into glucose and galactose? Option A, LAC-Y. Option B, LAC-A. Option C, LAC-Z. Option D, LAC-B. The correct answer is LAC-Z gene. So this is the LAC-Z gene that codes for beta-galactosidase protein or enzyme that breaks down lactose into glucose and galactose. Question number 7. What is the role of allolactose in the regulation of the LAC-operon? Option A, it acts as a co-repressor inhibiting transcription. Option B, it binds to the LAC repressor causing it to detach from the operator. Option C, it directly binds to RNA polymerase to enhance transcription. Option D, it's a product of glucose metabolism. The correct answer is it binds to the LAC repressor causing it to detach from the operator. So this is the allolactose. This allolactose binds to this lac repressor, causing the detachment of this lac repressor from this operator, thus promoting transcription of the structural genes. Question number 8. Under which condition does the lac operon exhibit the highest level of transcription? Option A, lactose absent, glucose present. Option B, lactose present, glucose absent. Option C, lactose absent and glucose absent. Option D, lactose present and glucose absent. The correct answer is lactose present and glucose absent. In the case of lac operon, this E. coli uses glucose as the primary nutrient source. When glucose is absent, lactose is used as an alternative nutrient source. Question number 9. When glucose levels are low or absent, ATP synthesis reduces and some ATP is converted to CAMP. What does CAMP then bind to in the context of lac operon regulation? Option A, lac repressor. Option B, RNA polymerase. Option C, CAP or catabolite activator protein. Option D, allolactose. The correct answer is CAP or catabolite activator protein. Question number 10. What is the effect of CAMP CAP complex binding to a specific site near the lac operon promoter? Option A, it inhibits RNA polymerase binding. Option B, it helps RNA polymerase bind strongly to the promoter, enhancing transcription. Option C, it causes a lac repressor to bind to the operator. Option D, it directly breaks down lactose. The correct answer is it helps RNA polymerase bind strongly to the promoter, enhancing transcription. As you see here, ATP is converted to CAMP, that CAMP level rises and CAMP binds to CAP or catabolite activator protein, and this complex, CAP-CAMP complex, bind to CAP site near the promoter, enhancing transcription. Question number 11. Consider the tripoperon which synthesizes tryptophan. When tryptophan is abundant, it acts as a co-repressor. What effect would this have on the tripoperon? Option A, it would bind to the trip repressor, causing it to detach from the operator. Option B, it would prevent the trip repressor from binding to the operator. Option C, it would bind to the trip repressor, allowing it to bind to the operator and inhibit transcription. And option D, it would enhance the transcription of tryptophan synthesizing genes. The correct answer is it would bind to the trip repressor, allowing it to bind to the operator and inhibit transcription. Question number 12. In the lac operon, why is transcription at a low level when lactose is present and glucose is also present? Option A, the lac repressor remains bound to the operator. B, allolactose is not produced. C, low CAMP levels prevent CAP from binding, thus not enhancing RNA polymerase binding. Option D, RNA polymerase cannot bind to the promoter effectively in the presence of glucose. The correct answer is low CAMP levels prevent CAP from binding, thus not enhancing RNA polymerase binding. So as you see, as a result of low CAMP level, there won't be complex formation between CAMP and CAP, thus not enhancing RNA polymerase binding or not enhancing the rate of transcription. Question number 13. What is the default state of the lac operon? That is when lactose is absent. Option A, on with high transcription. Option B, off due to the active lac repressor binding to the operator. Option C, partially on regardless of glucose presence. Option D, constitutively expressed. Correct answer is off due to the active lac repressor binding to the operator. As you see here, in the presence of glucose and in the absence of lactose, that is a normal condition, this lac repressor, this repressor proteins binds to this operator site, thus preventing the progression of promoter for the transcription of structural genes. So transcription is blocked, or lac operon is generally repressed in default condition. That's why it is called as negative regulation. Question number 14. The lac operon is an example of what type of gene regulation? Option A, positive feedback. Option B, negative regulation and positive regulation. Option C, only positive regulation. Option D, only negative regulation. The correct answer is negative regulation, that is due to the repressor protein that is binding to the operator, and also positive regulation by means of cap CMP complex formation, and also means of which that enhances the rate of transcription. Question number 15 and the final question. If a mutation occurred in the operator region of an operon such that the repressor protein could no longer bind, what would be the likely outcome? Option A, the genes of the operon would always be turned off. Option B, the genes of the operon would be transcribed constitutively. Option C, transcription would only occur in the presence of an inducer. Option D, the promoter would become inactive. And the correct answer is the genes of the operon would be transcribed constitutively, or it remains always on. If you have any confusion, you can watch our video on lac operon for better understanding. Hope you are benefited from this video. Comment your score below. Take care, stay blessed. Thank you so much. You are with biologyexamsforyou.com.